Thank you, dear organizers, for inviting us and for the wonderful opportunity to say a few words today about the INMARE project, a wonderful, very exciting new project that is about to start. As Niall already mentioned, this is a project about mining the marine environment for enzymes that can be used in industrial applications. Before I start, I would like to apologize for Peter Golishen from the Bangor University, who is the coordinator of this project and who could unfortunately not join us today. So he has asked me to speak on his behalf, and because we are also supporting Peter, my group, in the communication, outreach and dissemination of results, and I'm here in that capacity together with the colleagues from the University of Toronto by video message later on. Let me start by sharing a few basics about the project. So as I mentioned, we start this month. It's a 4-year project with a budget around 6 million euros. We have 23 partners from 12 countries, including Canada. As I mentioned, it is coordinated by Peter Golishen, and I really have to compliment Peter for bringing an excellent, a very complementary team of both industrial and academic partners together. There are more than around 30% of the partners are from industry. You can see the partners on the right-hand side of your screen. Before giving you a little bit more specific information about the project, I would like to take a step back and present INMARE in the wider context. INMARE is a project which fits into research which we call, or which we would consider blue or marine biotechnology, which in short involves the application of cutting-edge biotechnological tools on marine organisms or part thereof for developing a very diverse range of products and services. And among those products are enzymes and bioactives, which is really at the core of the INMARE project. But why are we looking into the marine environment for biotechnology applications? Well, for a number of reasons. Life in the oceans is very ancient, had a very long time to adapt to very different conditions that prevail in the oceans. We have a huge diversity, also partly through to the very extreme conditions at some locations, which you can see at the bottom of the slide. And this gives rise to adaptations to these conditions and production of a very diverse set of chemicals, biomaterials and bioactives and enzymes, which often have very unique properties which can be used for different purposes. This has led us to believe that marine biotechnology can contribute to addressing some of the most burning challenges of the 21st century in terms of developing new drugs, improving aquaculture processes to improve the supply and the quality of the food that we have at our disposal coming from the marine environment, trying to develop alternatives to the fossil fuels, notably through algal biofuels, but also in a way to better manage the marine environment. We can think of applications where we use marine microorganisms or processes to clean up oil spills, for example. And finally, also, marine biotechnology can help us develop new industrial products and processes, which is really at the core of the INMARE project. Why would we be looking at enzymes from the marine environment for these industrial processes? Because enzymes are more specific, they require less energy, and also use much less environmentally damaging products in the process than chemical synthesis processes do. And for these reasons, using enzymes in biocatalysis will be much cheaper and offers a greener and safer alternative to classical approaches. The huge potential of marine organisms for biotechnological applications has been recognized since a long time on both sides of the Atlantic in numerous strategic publications and think tanks. More than 10 years ago, for example, the National Academy of Science in the US has published a report addressing the the challenges and opportunities for marine biotechnology in the 21st century. In 2008, later on, the EC-US Task Force on Biotechnology met to discuss the challenges for marine genomics and the opportunities for research. The European Marine Board has also played a very important role to sketch and make recommendations for the framework and strategic development of marine biotechnology in Europe. And more recently, the Director General for Maritime Affairs and Fisheries has recognized marine biotechnology as one of the 5 blue growth sectors which show a great potential to support the maritime economy in the future. So where will we be looking for all of these new applications and new enzymes? Well, naturally, we will be looking where biodiversity is greatest. And then we turn to the microbial world, which really, where microbes really dominate the biosphere. You can see the ellipse where the microorganisms are situated on the tree of life. It is not a surprise, therefore, that today the majority of the applications in biotechnology are microbial-based. There is one major drawback, though. There is only a small fraction of microbes that occur in the environment, which we can actually cultivate, isolate and culture in the lab to study and to see what kind of properties they possess. And that's a major drawback, classically. But what can we do? The genomic revolution has provided a number of solutions and tools to circumvent this, the necessity to culture microorganisms in the laboratory. Notably, through metagenomics, we are now able to extract genetic material from the environment, from mixed samples, and use that genetic material in different strategies to express the genes in host organisms and analyze the resulting proteins that are produced. Or we can sequence the genetic material and analyze it, analyze the data based on the knowledge that we already have. In spite of the huge potential and the technological advances and the knowledge in bioinformatics that we have gained over the last 10 years, in reality, today, there are still very few marine enzymes which make it to the market. There are many challenges remaining, and they are listed and identified by the scientific partners in the project. I won't take you through it. You'll find all of those details and more information really on our website, and invite you, it will be launched very, very soon to have a look. But what I can tell you about INMARE itself is that the project will really look at the main bottlenecks along the value chain, trying to streamline the enzyme discovery pipeline, and trying to reduce the current timeline from 7 to 3 years. It will specifically try to do that by working on 2 steps which are very labor-intensive and very costly, notably the expression and optimization, which basically is a step which involves making enzymes more stable and more performant in an actual industrial setting, which is very harsh conditions. What is the strategy to do that? To shift attention more upstream at the beginning and try to screen for protein varieties which actually already show promising functionality at industrial conditions, so using very innovative screens. On the right-hand side, you can see a schematic overview of the workflow of the project. Without going too much into detail, I can tell you that the project will apply cutting-edge tools, state-of-the-art facilities, and complementary expertise of the partners to really address the main challenges along the pipeline. And as a starting point, the project partners already possess a very large number of gene libraries, source material. In addition, there will be sampling of several marine environments, notably extreme environments that have not been investigated in the past, and also involving sampling in the Atlantic. The project will work across all technological readiness levels. Main focus will be on levels 1 to 6 with the development of new tools and techniques, mainly screening platforms. But there will be also a lot of attention by the industrial partners on the levels 7 to 9, where enzymes and prototypes will be brought into realistic industrial setting to demonstrate the feasibility. It is important in this context to highlight that INMARE is not a project that stands on its own. It's really building on knowledge gained and materials gained during years and years of collaborations in a number of other European projects, national projects, and international initiatives. To conclude, to say a few words about the Atlantic dimension, as I mentioned, the Atlantic is an important resource for also the INMARE project as a source for microbial genetic resources. The coordinator has asked me to share a few lessons from previous research collaborations that the partners were engaged in, also involving partners from other regions outside of Europe, where they show a very high added value of this level of collaboration, but also that it's very difficult to bring non-EU partners on board. And as you know, this involves serious efforts in terms of synchronization and application of matched funding. Finally, in terms of contributing to the Galway statement, the project brings on board a collaboration with the University of Canada, but also we are working with an international cluster of companies and universities, which is called CLIP 2021, which also includes groups from Brazil, Canada, and the US. So hereby, I would like to end my part of the presentation, a short overview. And I've been told that if I switch to the next slide, I will have the opportunity to start a video from the colleagues from the University of Toronto.
My name is Alexis Sevchenko. I'm a structural biologist, and I'm a research associate professor at the Department of Chemical Engineering and Applied Chemistry here at the University of Toronto.
I'm Alexander Yakunin. I'm research associate professor in the same department, Chemical Engineering and Applied Chemistry at the University of Toronto. I'm an entomologist working on enzyme discovery for industrial applications, including biofuels, bioenergy, biocatalysis, and bioremediation.
Yes. So we're here at what we call the Biozone Center for Applied Bioscience and Bioengineering. This is the common research space that our department has created several years ago that unites 9 laboratories in our department, that unites people that look at the same problem from different perspectives, from enzymologists, structural biologists, as well as process engineers and microbiologists and bioprocess engineers, that we need all these guys, all these people, all this expertise combined to address the complex issues that's facing our civilization in terms of getting streamlined the industrial application and getting new biotechnological application in place. So in this respect, we're very excited to tell you that about the INMARE project and what would be our involvement in that. But first of all, congratulations to Peter Galichin and his colleagues to be able to convince to the reviewers to fund this project. I think it's great opportunity for all of us to work together and to work on this project. We worked with Peter and his colleagues before this collaboration happened, not overnight. In fact, for 5 years, Peter was leading another ES-funded project called Mamba, and we were part of that as well. And the good thing was that it also coincided of us obtaining Genome Canada grant of similar application. So instead of duplicating some of the efforts, we were able to enrich both projects with complementary expertise that we have on both sides of the Atlantic. And I think it worked really, really well, that we were very happy to do it, that we were very happy to continue on this path, because I don't think any of our labs would have been able to accomplish what we did in 5 years alone. So we're looking forward to work with Peter and his colleague on mining the richness of environmental microbiological libraries that Peter is so good on creating and distributing. And we can bring our expertise in molecular mechanism of protein function as well as enzymology. Maybe Sasha wants to say something.
Yeah, so before we already made great contribution to MAMBA project. It solved many protein structures, identified thousands of new enzymes, and we believe that the continuation of this work will produce even more important information about new enzymes and new activities, and we will be able to develop even more industrial application. So from our part, we are also interested in this project because we are preparing another grant application from Canada. So we would like to match that our activity and our funding with the INMARE project, and then pull together all these resources and get exciting new results. Yeah.
Again, congratulations to Peter and everybody who contributed to INMARE. We're very excited about being part of it. I'm looking forward to work with everyone. And again, the main message is, it's great to have an opportunities to work together on both sides of Atlantics to the common goal of getting new science into industry. Thank you very much.
Thank you. Bart.